All right, good morning, everyone. Last time we talked about this whole process, simplified, going from DNA sequence coming raw off of a sequencing machine, aligning, and then using a variant detection tool like GATK or Freebase to get a VCF file. And just as a quick reminder, the VCF file, or variant calling format, all it's reporting is all the sites in the genomes of the individuals that you told the variant caller to look at that are in BAM format. It's going to report all the sites where those individuals have a different allele with respect to the reference genome, at least one different allele. And remember that it has to make that decision based upon the evidence for and against genetic variation. So is there enough evidence in the sequencing reads above and beyond what you might expect from sequencing error? Okay, so we get this VCF file, which is just yet another text file. Lines in the file are positions in the genome where those individuals that were looked at are different than the reference genome. And the columns indicate sort of natural things like the chromosome position on which that variant was observed, the reference allele, what the alternate allele is, what the genotypes of the individuals are predicted to be at those sites. And so the focus of today's lecture is on the fact that actually this VCF file, as it comes off of a tool like GATK or Freebase, is useful in the sense that it is reporting all the sites where there is genetic variation, but it's not all that useful because this VCF is what I like to think of as it's naked. It gives us there's no context. We can't just scan through this file. Well, maybe you can, but I couldn't scan through a VCF file and know, oh, that variant at position 1,749,651 on chromosome 7, oh, that's the third exon of a gene that I'm interested in. That must be the one I'm looking for. So there's not a lot of information in the info field of a VCF file to help give that context. So what we're faced with often when trying to use VCF files in the context of disease, which is sort of a common use case, we're left sort of doing this. We've got this huge file with millions and millions of variant lines in it, and we don't really know where to look. Which is at this position, is at this position that's relevant. And so what we really need is annotation. We need to annotate these VCF files with extra information that help us figure out which of the variants in that VCF file are actually potentially related to the phenotype that I'm studying. And in future lectures, we'll go over a lot of these resources. Hopefully some of these ring a bell. UCSC Genome Browser is an obvious one. It's a clearinghouse for all sorts of annotations for not only the human genome, but lots of other model organisms. I won't go into detail on all of these, but essentially this is the collection of information that we really need to put this one site that it seems to be polymorphic in the genome among these individuals in context. So the case study that I'm going to focus on today is something that I do in my lab and a lot of labs here at the University of Utah do and elsewhere, which is essentially using DNA sequencing. We're going to focus on exome sequencing, but it's also applicable to whole genome sequencing of a rare disease case. So in this case, dad and mom are unaffected. That's why they're unshaded in this pedigree drawing. The child, let's imagine, is affected with a rare developmental disorder or a monogenic disease, also known as a Mendelian disease. I don't think we've talked too much about exome sequencing, but the whole idea is that you're using the billions of sequencing reads that you get from a single Lumina run to focus on the 2% of the genome that actually encodes proteins. So oligonucleotide probes are designed that are complementary to each of the exons in every human gene for the most part. We hybridize bulk DNA to those probes first, so we enrich for that 2% that encodes proteins, and then we put the remaining DNA on the sequencer so that we get really, really deep coverage at exons. So we have the power, if you remember back to the coin flipping exercise, so we have the power to detect heterozygotes. Were they there? So the data in IGV from an exome experiment looks like this. We get super deep coverage at exons, mostly nothing in introns. And were this whole genome, we basically have roughly uniform coverage across this whole region. Okay? So this is the study design. We've sequenced the exomes of dad, mom, and kid. And that's why we might get a VCF file that looks like this, right? There's 3 different genotypes, one for mom, dad, and kid at each of the polymorphic sites. And just to set the stage, one of the things that we know from lots and lots of exome sequencing studies done here, all over the world, one of the largest studies is from this group called DDD, or the Deciphering Developmental Disorder Study. And the take-home message from this is that it's this sentence. We estimate that 42% of our cohort, their cohort are individuals with extreme developmental disorders. They carry pathogenic DNMs, which stands for de novo mutation. So it's a mutation that occurred de novo in either dad's sperm or mom's egg, encoding sequences. And approximately half of those de novo mutations disrupt gene function. So the take-home message, I mean, this is a really beautiful study. It's empowered by looking at lots and lots of trios, mom, dad, kid trios, like I just showed you. They basically find a lot of these developmental disorders are caused by spontaneous mutation in that 2% of the genome that actually encodes proteins. And they find an enrichment even in the subset of those genes that actually, are crucial in development. Okay, so I think that helps set up what we might be looking for. I mean, we sequence the exomes of dad and mom and kid. We get the FASTQ file, we align it, and we run GATK or Freebase to find all the genetic variants among those 3 individuals. Yeah?
What's the difference between disrupt and altered? It says half disrupt gene function and the remainder result in altered protein function.
I think what they're talking about sort of the difference between complete loss of function versus modified function. Right. So we've exome sequenced these 3 individuals. Now we've got, if you exome sequence mom, dad, and a kid, you typically get about 25,000 genetic variants that overlap coding sequence in a VCF file. So if our hypothesis is that it's a de novo mutation that is causing this developmental disorder that we're studying, we're essentially trying to figure out which one of the genetic variants in that VCF file underlies the phenotype out of the 25,000, right? So it's a bit of a needle in the haystack problem. Right, so to disrupt gene function, there's a bunch of different types of genetic changes in coding sequence that have different outcomes, right? I mean, this gets back down to central dogma. We could have a situation like this where the 3rd position in the codon is changed from a G to an A, but that codon CAA encodes the same amino acid as CAG, so this is the so-called silent or synonymous change because the amino acid sequence doesn't change. We could, in this case, the 2nd position is altered in the codon, and so this change from a CAG codon to a CGG codon actually does change the amino acid sequence. So that is a so-called non-synonymous or missense change, and that could potentially alter the function of the protein, the stability of the protein, and potentially what it does if it affects an interaction site or a serine, which might underlie stability or some sort of activating site that could actually totally change the function of the protein. In this case, we have a change from a C to T that actually alters this from a codon that encodes an amino acid to a stop codon, basically saying stop translation. That would be bad. So that would abruptly stop the translation of the resulting protein, and obviously affect it if it was sort of early in the transcript. And then another option is we could actually lose that stop codon, so it keeps translating through to downstream codons. So there's a whole spectrum of genetic changes in coding DNA sequence that can have lots of different potential implications on the structure or the function of proteins. And depending upon what the protein is and how critical that protein is in development, it may or may not lead to the phenotype that you're studying. There's a so-called set of loss-of-function genetic variants. So we just looked at one, nonsense. This actually leads to a premature stop codon. So basically, the resulting protein will stop here. We can have a frame shift. So if there's an insertion or a deletion of nucleotides that are not a multiple of 3 or out of frame with respect to the triplet reading frame, we can actually disrupt the amino acid sequence of everything downstream of that insertion or deletion. If we disrupt a canonical donor or acceptor site, that can affect splicing, and if splicing is altered and an essential exon that encodes a particular domain in a protein is removed or a new one is added, that can totally affect the resulting protein. And obviously, if we delete entire exons through so-called structural variants, which we'll talk about more next time, or delete the entire gene, obviously there's potential for a phenotype there, right? Because the whole protein might not even be made anymore. So these would potentially eliminate the function of the protein and lead to a phenotype. So the problem is that variants like this that lead to putative loss of function of protein exist hundreds of times in every one of our genomes. And part of this is the fact that we can computationally predict that a genetic variant may lead to loss of function in the protein, but we don't get to do sort of the functional validation of every one of those predictions in every one of our genomes. So obviously, if you have a variant that disrupts the function of one olfactory receptor gene in your body, you're probably going to be okay. So we maybe have lots of genetic variants like that, but there's a lot of redundancy built into our genome in terms of the pathways that are relevant to development versus things like being able to smell certain scents better than others. But if you look at this table, this is from a review that I wrote a couple years ago. What we're looking at as rows here are different world superpopulations, people of European ancestry, African ancestry, from the Americas, South Asian ancestry, or East Asian ancestry. What I've done is looked at the total the median number of coding variants observed in individuals from these different ancestries, broken down by different categories. So a typical, let's just look at one example, a typical individual of European ancestry has about 7,000 sites in their genome, which lead to a synonymous change in a heterozygous state. So one of the two alleles that they inherited from mom or dad is a synonymous change. And there's about 4,000 that were they inherited that change from both mom and dad. So we wouldn't, in the case of a rare disease study, we probably wouldn't be too interested in synonymous changes anyway, because it doesn't alter the amino acid sequence. So given the severity of the phenotype, we might say, well, that's probably not terribly interesting. There are definitely exceptions to that rule, though. But if we move out here into the putative loss of function changes, a typical individual of European ancestry has 93 coding changes that would lead to an addition of a premature stop codon in a heterozygous state and 35 in a homozygous alternate state. Now, probably the vast majority, if not all of those on average, affect proteins that aren't essential during development, right? And that's part of the problem. If you sequence any one of us, if we did an exome study of any of us and our parents, and we did an exome study of that child with a developmental disorder and their parents, the numbers of things that you're going to see are going to be very, very similar. It's just that one smoking gun that you're looking for, and the challenge is how do we find that needle in this haystack? And part of it is we want to use magnets, right? We want to use the right tools to sort of figure out where that needle is. And that's a little bit of what I'm going to talk about today. So the first class of tools that we end up using are effect predictor tools. They take a VCF file, this naked VCF file, which is all the positions in the genome where the mom, dad, and kid are different than the reference genome. And they take transcript information. So on sites like Ensemble and the UCSC Genome Browser have annotation files that report the position, the chromosomal position of every protein coding gene in the human genome, the start and stop positions of all the exons for all those genes, the start and stop positions for all the introns, etc., and the strand, the strand of the coding frame. So these tools like SNP-F, this one I'm mentioning here, and Anavar, take the VCF file and that annotation of where all the protein coding genes are, and they do the really complicated logic of figuring out, okay, at this position of the genome, it changed from a G to an A. What effect does that have on this isoform of this gene? Many genes have multiple different isoforms, so it has to make do that analysis. If there's 17 different isoforms for one gene, it has to do that prediction for every one of the 17 different isoforms. And that, I mean, given that it's basically just following central dogma, DNA gets transcribed into RNA, and RNA gets translated into an amino acid sequence, you'd think that would be fairly straightforward, but it turns out that it's actually kind of a hard problem. One of the most widely used tools for this is something called VEP, Variant Effect Predictor. This is developed by the group called Ensemble in the UK. And they've got a big development team that really does a good job making this work fairly well. But the point that I want to emphasize today is that there are many different tools to do this. VEP and Anavar, and there's one developed by the Yandell lab here called VAT, and there's another one developed by the Gerstein lab called VAT. Lots of different software. Software is written by people. People make mistakes. People make different decisions about what the software should do in different scenarios. So therefore, each of these different tools, given the same information, might make different predictions about what the impact of a given genetic change is on the resulting protein. That's one side of the problem. The other side of the problem is that there are many different descriptions of gene annotations. If you go and look at annotations for P53, which you might imagine would be sort of one of the most well understood proteins in the human genome, if you go look at each of these three different gene annotation sets, the number of isoforms and the exact structure of those isoforms for even P53 might be slightly different. So therefore, if you make predictions of genetic variants based upon one set of transcript annotations versus another, you might also get different predictions. So this leads to sort of the depressing state of affairs. Things have gotten better since this paper was published, but this is this paper from Davis McCarthy in 2014 made kind of a big splash and made people really sad for a while, because what it looks at is the predictions made by these two tools, Anavar and VEP, or the two combined, and asks how often given the same input, the same VCF file, everything, how often their predictions match. And only 2/3 of the time do the predictions actually match, which made me very scared at this time, because we were actually using these tools for some studies we were doing of unsolved cases of Smith-Magenis syndrome, and we were worried that the solution was hiding in plain sight, but we were missing it simply because we were using one set of annotations or one annotation tool. So we're wondering, oh gosh, do we really need to run every one of these tools with every one of these different annotation tracks to figure out all the potential solutions? Okay, so why might it be so bad? This is a figure from Davis's paper that I think is a really nice example. It's a bit busy to look at, so I'm going to try and walk you through it. Okay, so we've got coding sequence in this direction, on this strand, and the genetic change that's observed, say, in the kid that we're studying is an insertion of a single A nucleotide right here between this G and that A. Okay? This is the codon that change falls in. So if you were writing software to predict what the impact of that single A insertion is, there's many different options, right? It's an insertion of one nucleotide that is within the codon. So that, by definition, leads to a frame shift. It shifts the reading frame by one nucleotide. So you could say, all right, if I see this, I'm going to write the code, and it's going to return frame shift, and that's going to print to the file. You could also, because TGA encodes a stop codon, the insertion of this A, you could say, is a gain of a stop codon. So you could report it as a stop gain change. But the codon was already TGA, and you're inserting an A. So you could also say, well, it's synonymous because the codon is the same. It was encoding a stop codon, and now it still is encoding a stop codon. Or you could, the last choice is, you could really choose any one of these. Each of these is a sane prediction. And it's sort of corner case scenarios like this that lead to this discordance between the different software tools. Thankfully, subsequent to that paper, the developers of all these different tools started using a common set of rules and a common language called the sequence ontology, in part developed by Karen Eilbeck's lab here at UU. And that really helped to establish a common standard, a common language, so that the overlap in predictions by all these different tools is much higher, and they all put their heads together and really thought through like what the actual rules should be, so the predictions are better too. Okay. All right. So this would be this is an example of the complexity of making these predictions for one isoform of one gene. The problem that this review that Karen and Mark and I wrote a couple years ago addresses is that, well, for a given site, there's 5 different genetic change sites, A, B, C, D, and E listed here. You can see that these variants overlap multiple isoforms. So in this case, there's 3 different isoforms of this gene CCDC113. I forget why we chose this. But what you can see is that this second isoform of this gene lacks an exon here. So that exon is removed during splicing. So the position of the genetic change for A is right here, but the prediction for these 2 isoforms, the top and bottom, is going to be different than the predicted impact of that variant on the middle isoform, because it's not coding sequence in the middle one, right? Similarly here, these 3 different variants actually overlap 2 different genes. So for variant C and D, it overlaps this exon of CCDC113 and a different exon of this other gene, PRS-S54. So you're going to get 5 different predictions, 2 for the 2 isoforms of one gene and 3 for the different isoforms of another. So essentially what you end up having to do is look for every variant explore all the different possible impacts of that variant on different genes and the isoforms of that gene. And that's especially important because you might actually find that a genetic variant affects a gene that makes sense to you, but the loss of function impact of that variant only affects one isoform of that gene that actually isn't expressed in the tissue that's relevant to the phenotype. So that's the next level. You have to dig deeper and deeper into, well, where is this thing actually expressed? Is it, okay, it's only expressed in one tissue, and that tissue has nothing to do with where the phenotype that I'm studying manifests. So it's probably not a good candidate. So this is just another example of why this is complex. There's another fundamental truism in this type of analysis that I think is worth spending a little time thinking about. And it's sort of, there are many ways to state this problem, but I think in science in general, the more interesting that something looks, the more likely it is to be some sort of artifact and not a real biological signal. So the way that manifests in this kind of study is that let's say on the x-axis here is positions along a chromosome. And there's some level of error in our sequencing experiment. Some of it comes from base calling errors made by the sequencer, and some of these errors come from, say, just problems properly aligning the sequences along to every position in the genome. So there's this sort of low-level error rate that's fairly uniform across the genome. And then there's this other rate, which is the rate of true genetic variation in the system that we're studying. So in this case, in a proband, in a kid. And what this is illustrating is that true rate of variation actually drops in coding sequence, because coding sequence is going to be under stronger purifying selective pressure than sequence that's not in changes that are not in coding sequence. Does that make sense? Because changes in, say, an intron don't have nearly the impact as they do on an exon. So when this true rate of variation declines and approaches the error rate, that means by definition that most higher fraction of the things that we think are interesting in coding sequence are actually going to be errors. So this broken down into pie charts, proportion of things that are errors is represented as the red piece of the pie. It's low, relatively low in non-coding sequence, but that proportion increases in coding sequence because the signal and the noise are so close to one another. So another way to think about this is if your error rate and the rate of the thing that you're looking for are the same, then by definition, every one of your half of your predictions will be false, right? So it basically means when we see a really interesting variant in coding sequence that disrupts, is predicted to disrupt the protein, we need to really have some suspicion about that. We need to essentially look at every one of those types of variants by eye to make sure that it really, really passes, the sniff test. Another figure from Daniel MacArthur's paper about this, basically they were looking for loss of function polymorphisms in human protein coding genes. And one of the things that they find is that there's this enrichment of raw frame shift variants towards the carboxyl terminus of protein coding genes. And the idea would be that maybe those aren't so interesting. They're predicted to be loss of function, but if you have a frame shift that only affects like the last two amino acids of a polypeptide, it probably doesn't matter as much as if that frame shift occurred very early in the amino acid sequence and therefore perturbs 98% of the amino acids rather than 2% of the amino acids. So that's the other piece is you can have a predicted loss of function variant. You go and look at it, the sequence alignments supporting that variant in IGV, and you say, yeah, that looks pretty good. But then, and it's in a gene that you think might be interesting. But if that is at the very carboxyl terminus of that gene, then maybe it doesn't matter as much. Okay, so now I want to transition into another fundamental concept in this type of analysis. So developmental disorders are very rare. So if we find a variant in a gene in our proband that makes sense, would we believe it if we knew that that variant actually exists in 5% of human chromosomes? Would we still think, if we were given that information, would we still think it's interesting? Probably not if the phenotype occurs, say, in 1 out of 50,000 live births, right? So there's a huge disconnect between the frequency of the variant in the population and the prevalence of the phenotype. And if it occurs that frequently in healthy individuals, it probably doesn't actually lead to the phenotype. So that intuition has been around for a long time, but until very recently, we didn't have a large-scale study of DNA sequence from ostensibly healthy individuals. So the first study to do this was actually from Debbie Nickerson's lab at the University of Washington. They sequenced 6,000 individuals. And then about a year and a half later, this study from Dan MacArthur's group at the Broad Institute basically increased it by a factor of 10. So they sequenced 60,000 human exomes. Some of those samples actually are individuals with that had disease of some sort, but by and large, they are not individuals with rare developmental disorders. So these 60,000 individuals serve as a nice control population for developmental disorders. Yeah.
So I guess to me that like gets into language of what you're calling like which gene is actually responsible for the phenotype. Because like it could be that a certain variant is harmless in most cases, but like pretty harmful in like a certain context. Like maybe there's some other mutation, some other gene. But like when you're talking about like the molecular story about like what the protein does and in what tissue, like it's clearer for that gene that's a common variant. Does that make sense? What I'm saying?
I didn't follow the last part. It's clearer for that.
So like when you're trying to come up with the story for like why this protein and this dysfunction could like lead to this certain phenotype. Like it could be that common variant is like that's where like the more direct relation between like the variant and the phenotype is, but there could be like this other piece that's like really rare.
Yeah. So I think there's a couple things going on there. That could be sort of a predisposition allele for a more common phenotype rather than a really rare phenotype. Or it could be actually it underlies a rare phenotype too, but that's sort of like the first hit. You have this common variant segregating the population, but it requires a second hit leading to like a compound heterozygote for a rare disease phenotype. So yeah, it's not, I mean, the way I'm describing it is oversimplified, but this resource of 120,000 chromosomes really gives us the first piece of that analysis, but it's not as simple. It's just like, oh, how common is it in the population? So we talked about this before, but the vast majority of the variants that they found in this study were rare singletons. In fact, 50% of the 9 million variants that they found were present as a heterozygote in 1 out of those 60,000 individuals. This gets at that notion that there's this huge excess of really rare, extremely rare variation in the human population, primarily owing to explosive population growth over the last 2 millennia. But 5, 7 lectures ago, I don't remember, we talked about all the different processes that can alter the frequency of a given allele in a population. So I think the most intuitive one for me is migration. So you have a founding population, that's in San Diego, for instance, and the allele frequency in San Diego is 10%. But then, what is it? I'm probably the only one who watched Gilligan's Island as a kid a lot, but Gilligan's Island, they get, on the SS Minnow, and they get stranded on this ridiculous island, and now you've got a founder effect. And maybe the allele frequency of that small group is much higher than it was in the founding group in San Diego, and they go on and have kids, and the new population grows up on that island, and all of a sudden you have a very different, 20 generations later, you have a very different allele frequency on that island group. The same thing has happened worldwide. The same genetic variant has very different allele frequencies across different world populations because of things like migration that lead to population bottlenecks, randomness and genetic drift, and differential sort of mating behavior. Maybe there was a little bit of intrabreeding within a population as they migrated out. So when we think about the frequency of a variant in a population, it's sort of fraught with problems to think about a single frequency across the world population, because it can vary quite a bit. So this is a slide that I borrowed from Jessica Chong, and it focuses on one variant. It doesn't matter where this is in the genome. I don't think it's even tracked. But the overall allele frequency of this variant among the 1,000 Genomes Project is 4 out of 1,000 chromosomes have this alternate T allele, whereas 996 out of 1,000 chromosomes have a C allele. So that variant might seem, if the frequency of the disease phenotype that you're studying is roughly 4 out of 1,000 individuals have it, this might be an interesting candidate. But when you look at the details, it becomes less interesting, because there are certain populations, like in Finns, it's 5% allele frequency. In Europeans, more broadly, it's 1%. In people from the Iberian Peninsula, it's up to 3%. So that's much higher frequency than the disease phenotype. And so the intuition is, if an allele, a DNA change, leads to a phenotype and is penetrant, fully penetrant, every individual that has the allele has the phenotype, then this doesn't make sense, right? Because that penetrant should be applicable to people of any ancestry. Now that makes some assumptions about modifier alleles and things like that, but ignoring that, we could use the logic that, well, it's probably not a great candidate now that we know that it's so common in these other world populations, unless these other world populations have, along with this variant, another modifier allele that suppresses the phenotype introduced by this variant. And that's what's so important about not only sequencing lots of healthy humans to do this type of work, but sequencing lots of healthy humans across diverse ancestries. And one of the things you probably appreciate is most human genetics work is sort of very Eurocentric. It's mainly focused on people of Western European ancestry, loosely speaking. That's changing, thankfully. There's large-scale efforts in the U.S. and worldwide to sequence more diverse ancestries to sort of dig into the details of things like this. Okay, another example is much more striking, I think. This variant, I again lost it doesn't matter really where it is, but it's mentioned in this review. The average allele frequency of this variant is something like 1%. And that's because a lot of these other populations that were sequenced basically don't have it at all. It's never seen. But if you look in people of African ancestry, it's a very common variant. So just looking at an average summary statistic really belies the truth and the sort of the complex demography of this variant in the human population. Okay. What's really cool about this Nomad is now called Nomad. The 60,706 was called EXAC, the Exome Aggregation Consortium, and then they went even bigger and started sequencing genomes and exomes and rebranded themselves as Nomad, or the Genome Aggregation Database. It's a really amazing resource for human genetics. If you're doing any human genetics work, especially in rare disease, this resource is your friend. What's really nice is you can go type in any gene, and it'll bring up this list of all the genetic variants that are observed among those now 120,000 or 180,000 individuals, something like that. And you can hover over them, figure out where they are on the protein. They draw gene models for every protein. I chose ATM. Unfortunately, it has lots and lots of exons, so this is a bit of a mess. But were I to click on any one of these variants, what you get is something like this. It's a breakdown of how many homozygotes and heterozygotes were observed among all the individuals that they studied, broken down by their predicted ancestry. And so in this case, we can see, yeah, I mean, this variant is quite rare in people of East Asian ancestry, but is fairly common in Europeans, non-Finnish Europeans. Okay. Anyone have any sense of how you go about knowing what the ancestry of these individuals are? They actually don't know. They weren't told. People didn't walk in to this study and say, my ancestry is blah. Anyone have a sense of how you would do that? Yeah. That's right. Yeah. So there's these set of sites in the genome that are called ancestry informative markers. And as the name might imply, their frequency is a good whether or not you have a variant at one of these sites is a pretty good predictor of your ancestry. So they choose markers that are sort of really biased in terms of their frequency in different populations. But one variant isn't enough to make that prediction. It can be, could lead to a false prediction. But when you have thousands of markers like that, the information that you collect in aggregate is what gives you that really confident Bayesian prediction. So it really gets back to Bayes' rule. You have the prior probability of that variant being from an individual of a given ancestry, and then you compound all those observations into a really good posterior prediction. And that's essentially what Ancestry.com and 23andMe and all those do. They're looking at ancestry informative markers and making a prediction about your ancestry. Okay. Right. So in the context of rare disease, so far we've talked about what the predicted impact of the variant is, whether it lies in a gene, and whether it's heterozygous or homozygous. But obviously, it matters sort of the mode of inheritance. So when you're studying Mendelian disorders, you often know whether it's inherited in an autosomal dominant fashion or an autosomal recessive fashion. I'm going to skip X-linked stuff for now. But if you've not heard about these types of things before, the idea is in an autosomal dominant phenotype, individuals that are heterozygous for the disease-causing variant have the phenotype, and any offspring that inherit that allele, marked in, I guess, white square here, have the disease. So this would mean that this allele is fully penetrant for the disease phenotype. If you have the allele, you have the disease. In contrast, in an autosomal recessive case, you often have situations where both parents are unaffected. They're carriers. They're heterozygous for that variant. And using Punnett square stuff from high school, 25% of offspring will be affected because they're the unlucky ones that inherit the two deleterious alleles from mom and dad. Okay. So this is where everything we've talked about so far in the VCF file is like information, annotations, and positions in the genome. So we know like it's in a particular gene that matters, and we can look up the allele frequency of that allele. Now we're talking about the genotypes of the individuals to track whether or not that variant makes sense in a given gene, given its predicted consequence, given its predicted allele frequency. It only makes sense if the inheritance pattern is right. Right? Because in our study design, I think mom and dad were both unaffected, kid is affected. So we wouldn't be looking for sites where there's a heterozygote shared between the kid and mom and dad, because mom and dad are unaffected. Okay, so in a case where mom and dad are unaffected and kid is affected, there's two sort of simple explanations. One, it's an autosomal recessive phenotype. Mom and dad are carriers, and the proband is the unlucky one that got the two bad alleles. Or it's a de novo dominant phenotype. So that mutation occurred de novo on either dad's sperm or mom's egg. Kid has the phenotype because, well, the parents just didn't have the allele. And so in that case, we'd essentially be looking for situations where there's an allele that's present in the kid that is not seen at all in the parents. Because remember, we've sequenced blood from the parents. We didn't actually get to assay their germline. So there's a number of tools to do this. We don't have the time in this course to go through them. One is something that I wrote a while ago called Gemini. This is what we use in the lab quite a bit. Brent has written a new tool called Slivar to do this, but essentially it is taking VCF files that have been annotated with tools like VEP or what have you, annotated with the allele frequency, and then essentially you tell it to look for de novo mutations in the affected kid. And it knows the relationship of all the individuals in your VCF file and looks for variants that meet these different inheritance patterns. Okay. So the way Gemini worked, just to think through this for a second, is it would take a VCF file, which has all the obviously has all the positions of genetic variation in the genotypes, but it also requires this other file called a PED file. And a PED file describes the sex, the relatedness, and the phenotype status of all the individuals that are in the VCF file, because one of the shortcomings of the VCF file is you just have that column of genotypes which says sample name. But imagine you've got a study with like 1,000 different samples in it, and that's spanning 200 different families. You can't by eye, unless you have a really nice clever encoding scheme for the sample IDs, know the relationships of all those individuals. So the PED file describes that, and so given those two pieces of information, this tool Gemini takes all those variants and the known relationships, annotates the variants, so dresses up that VCF file with all this information that's required to make those predictions, and then it put it into a database that could then be queried to look for causal variants. There are other tools out there that do this. I mentioned Slivar. It does the same thing, takes a VCF file and a PED file and makes the predictions directly without having to use a database. Okay? But the key thing that I want to emphasize here is that this is software that is making predictions about candidate genes and candidate variants underlying rare disease by integrating all this extensive annotation that's required to make those predictions, essentially to figure out which of the 1, 2, 3 variants in an exome underlies the phenotype rather than scanning through all 16,000 and doing a Google search on every one of the 16,000 candidates. So one example that I would just want to touch upon is compound heterozygotes. So we talked briefly about this before, but I think this kind of gets at, I don't know your name, but the scenario you were talking about where, what's your name by the way? Marshall. What Marshall was talking about, where you could have one variant that's sort of at a fairly common in the population, but it takes a second variant to lead to the phenotype. And so the way this works is, imagine that we've got both dad's chromosomes here labeled as two different colors, one color for the chromosome that he inherited from his mother and one for the chromosome that he inherited from his father. Same for mom. But you notice that each of these parents are carriers. Mom has a deleterious T allele on this chromosome, and dad has a deleterious A allele at this site on his chromosome. So if a kid inherits this haplotype from dad and this haplotype from mom, which would happen with a 25% chance, then the kid is heterozygous at this site and at this site, but the deleterious allele exists on the two different haplotypes. So both copies of the gene are disrupted. in this case. But we only know that by looking at multiple sites in the genome. So we'd have to have some sort of prediction about the functional consequence of this variant, so maybe a missense change, and we'd have to know something about the allele frequency of this putatively deleterious T allele, and we'd have to have the same information for this A allele to sort of put together the puzzle to predict that, yes, this individual did inherit this as a compound heterozygote. We can know that directly by looking at parents' genotypes and figure that out, basically doing like Sudoku. But we would also need to know what the impact of these deleterious alleles is on the protein and how common they are. Okay, so couple of different things that make this challenging, and this gets to getting at phase genotypes again. So imagine these are two different sites in the same gene, and in this case, the G allele is inherited from mom quite clearly. So we can know unequivocally that G was inherited from mom, and so we put the G allele first, and then we use this vertical pipe sign to say it's a phased allele. And in this site, the T is clearly coming from dad, so we can say that this is obviously the paternal allele. Okay, so this is a high confidence compound heterozygote example because we know by inspecting parents' genotypes that both of these alleles were inherited on different parental chromosomes. Okay, I'm not going to walk through all these, but here's an example. This other example is a little more typically what we see, where you have a deleterious allele at two different sites, but in both cases, that deleterious allele is inherited from the same parent. So it's only one of the two chromosomes that's disrupted. So this would not be a good recessive case because we can clearly see through the inheritance of these alleles that only one gene copy is messed up. Okay? There are different tools for doing this. This is how you would use Gemini. You basically say, hey Gemini, this is a Unix command. Run this tool that looks for compound heterozygotes. And then there's lots of different things that you can do to increase the stringency of the candidates that it returns. Like for instance, we can make sure that it's rare. So this maximum alternate allele frequency across all individuals from that Nomad study says, well, I only want to look at compound heterozygotes where both of the alleles are present on less than 1 out of 1,000 chromosomes across all ancestries in that Nomad study. So to really hone in on the high confidence things. So we use this pretty extensively here at the University of Utah, and it's been really fun collaborating with some of the other centers for Mendelian Genomics to develop this tool. It's now pretty widely used in rare disease research. So if you're doing this type of work or any family-based studies, this piece of software as well as Slivar and some other one, actually a lot of the relevant software is developed here at the University of Utah. I want to touch upon two other pieces of, well, one other actual piece of software from the lab that Brent wrote. The idea is that you have these sort of undressed or naked VCF files from any variant caller, Freebase, GATK, Mutec, no matter what you're doing. Tools like VEP and SNPF will do a good job of predicting the impact of all those genetic variants on genes. But there's usually a lot of other information that you might like to bring in, like allele frequencies from the 1,000 Genomes Project or Nomad, whether it's a known variant, disease-causing variant in a database like ClinVar, et cetera, et cetera, et cetera. Like, is it a variant in a gene that your lab is really interested in? Imagine you have a list of those genes in bed format. This tool called VCF Animo is just sort of a general purpose way of taking an input VCF file and a collection of lots of other annotations so that every one of the variants that's in that VCF file gets embellished with annotations from all these different files that you give it. And that information gets packed into that info field, which is, I don't know, the 7th or 8th column in the VCF format. So it just slams the information into that file, into that field, so you can grep for that information later on if you when you're looking for relevant annotations. So concrete example, this is the VCF file before VCF Animo. So the only information that's in the info field is like how many chromosomes had this alternate allele and what's the total frequency of that alternate allele. We can give it this little configuration file that tells it which files we want to annotate this VCF file with. So a VCF file from the Exome Aggregation Consortium, maybe dbSNP, which tells us all the known genetic variation, and maybe some measurement of conservation. Then for each of those files, we tell it what fields we want to pull out of those files and what we want to name the resulting annotation in the info field of this original VCF. So the after is essentially we've got all these new annotations, and each of these new annotations in the info field connect back exactly to these name fields that are set up in this configuration file. So it works with any species as long as it's a valid VCF file and you have good annotation files, you can annotate your data pretty easily. Okay, so that's it for today. I just want to, hopefully you got my email. I apologize for the problems with homework 5. I know some of you have started messing with that, and I neglected to actually give you the data that you needed. But it should be there in your home directory now. If you don't have that, please email me. I think I got it all working right. And just as a reminder, homework 5 is due next Thursday, I think, the 27th. Anyone have any other questions about homework, issues? Everything going okay? Yeah.
I tried running the freebase command before you start homework 5. And it said permission denied to read the VCF file. Oh, you have to copy it out of homework 5 into your own directory. So once I copied it, it worked.
Copy.
But I
Can you
can't do it in the homework 5 folder. I can copy all those files into my own folder.
Oh, I can change that for everyone. There's just a permissions problem on it. So you could write the resulting file to another directory if you want, or I can try and change the permissions for everyone. So basically, just to explain what I did, I created a homework 5 directory in my workspace, made all the files, and then basically wrote a command to copy that whole directory and all the files to each of your home directories, and it just copied the permissions that are associated with that directory in my space. So the other thing you could do is just change the permissions of that directory using that chmod command. So you would have to add write permissions, so that's +W to that directory, and it should work. Okay. But if you have any problems with that, I'll try and change that for everyone now. But if you have problems, let me know. Okay. All right. Thank you.